Hi, my name is Ava Bender, and I am Dr. Bender's helper. Today we are going to learn about feedback.
Like Ava said, we're going to talk about feedback tonight. So feedback basically is our coordination of all of our systems, all of our mechanisms working together and being able to respond to the environment. It's based off of it's the timing and the coordination of your biological mechanisms that depend on the organism responding to the environment. So basically, this is the coordination of your body's ability to, and we're think multicellular right now. That would make this a little bit easier, I think. So let's think in terms of your body. This is your body's ability to grow, to reproduce, to maintain homeostasis. It's all of those things, all of those systems working together to be able to maintain that for you. And it's all based off of responding to the environment, both internal and external stimuli. So feedback mechanisms are going to be categorized in two basic ways, either negative or positive feedback. And we're going to look at both of those in detail. So we're going to look at negative feedback mechanisms first. So there's not really a lot of new stuff here. What is key here is that you need to be able to apply the different parts and the concept of a negative feedback loop to potentially unknown situations, and you need to be able to write about a negative feedback loop. So we're going to pick one particular example and follow it through, and we'll work some with it in class as well. So negative feedback loops, remember, they're not a bad thing. They are actually a very good thing. Okay? They offer regulation for your body. These are what help maintain homeostasis. So they're negative in the sense that they're going to do the opposite. They are returning your systems back to their normal target range. Think body temperature, right? There's a target range that your body temperature likes to be. When it gets too hot, you kick in certain systems that help you, sweat and help you try to cool down. When it gets too cold, you kick in other systems that help you shiver and help redirect blood flow to try to warm you back up. So it's returning the system back to its target range. So it's turning things on and turning things off. Okay? These work at both the molecular and the cellular level. So the various concentrations of molecules respond to these, and then the cells themselves also respond with these negative feedback loops. So the example we're going to kind of look at a lot is blood sugar regulation. We talked about insulin already in class a little bit with our example for a hormone. So our diagram over here on the right is going to help us kind of walk through this. So here in the middle, we've got what is considered homeostasis. And you'll notice that's a range. So your body likes between 90 and 100 milliliters of around 90 milligrams of glucose per 100 milliliters of blood. Okay, and that's an around situation. It's not an exact. It's not like if it goes to 89, everything kicks into gear, or it goes into 91, everything goes crazy. So after eating, so pretty much after eating anything, unless you're on a very, very strict no carb, low carb diet, after eating, your blood glucose levels, they're going to start going up. Right? Because you've taken food in. And so you're digesting that food, releasing that, and you're releasing those parts that the food is made out of, and then your intestines are absorbing that into your bloodstream. So your blood sugar is being monitored, and the blood glucose levels start rising. And so when that signal happens to start rising, that's going to trigger the pancreas to release insulin. Insulin is a hormone, and so that insulin is going to go affect target cells within the body. So the beta cells of the pancreas will release insulin, and it's going to impact almost every cell in your body. The only cell in your body that's really not going to respond to insulin are the cells in your brain. Otherwise, pretty much every cell in the body is going to start taking glucose up from the bloodstream. And as we mentioned before, the liver is going to be a big one that's going to do that, as well as skeletal muscles. So the liver and skeletal muscles are going to help take that excess glucose, and then they're going to convert it into glycogen. If you remember, glycogen is a polymer of glucose. It's how animals store their extra glucose. So the insulin's going to bind to the receptors that are on the membrane of the cell. In particular, let's talk about the liver. So the insulin will bind to the liver receptors. And then the activation of those receptors are going to trigger that signal transduction pathway. So that's going to amplify the signal so that the cell can have a response big enough to make a difference. So that cellular response now is it's going to insert a particular kind of receptor. It's called a GLUT4 receptor. So now it's going to insert these GLUT4 receptors into the cell membrane. These GLUT4 receptors allow glucose to get in. They allow more glucose to come into the cell. Okay? Therefore, then the cell will start to synthesize the glycogen and it will start to produce proteins. And so my end result is my blood sugar level is going to go down. They're going to lower back down into that optimal range. Okay? So that's one part of the feedback mechanism. It gets too high. The pancreas, the beta cells in particular, will release insulin. Insulin will go to these target cells, which will be all over the body, mainly liver and skeletal muscle cells. The insulin binds to the receptors. The activation of those receptors triggers that signal transduction pathway. So the response then is the insertion of those GLUT4 receptors, so more glucose goes into the cell, synthesizing glycogen, lowering, and then that causes the overall response to the body, to the organism, the overall lowering of the blood sugar level. It can also work the other way, right? Our blood glucose level could go down. We haven't eaten in a really long time. So now the pancreas, in this case, it's the alpha cells. So we have the beta cells secrete insulin and the alpha cells secrete glucagon. Glucagon is released from the alpha cells of the pancreas. The glucagon now will go to these target cells that have stored the extra glucose. So for instance here, the liver. The liver has that glycogen stored in it. So the liver is going to have receptors that will respond to glucagon. So the glucagon is going to trigger a response in the cell to break the glycogen down and start releasing glucose back into the bloodstream. So now I get an overall organism response of having a higher blood sugar level. But that all came from those cellular pathways and those cellular responses. So that would be an example of our negative feedback loop. Let's look again real quick at let's review, because hopefully you remember these from before. Let's review our different parts of a feedback mechanism. So the first part is going to be, right, we've got our stimulus. So in this case, our stimulus would be those blood sugar levels. Is it too high? Is it too low? Okay, so from the stimulus, right, that information is going to go to the integration center. That would be the brain. The brain would then say, okay, I need to release some insulin. So now the insulin gets released, and now the insulin can carry out the final response. So the effector would be the pancreas here. So my stimulus is my blood sugar levels. My information gets processed to make the determination that it's too low or too high, and so I need to either release insulin or release glucagon. And then the overall response is going to be the change in the blood sugar level. So that's our big picture response. But again, at the cellular molecular level, I have insulin communicating with the cell, telling it to put more receptors in so it can take in more glucose, or glucagon binding with the cell, telling it to break the glycogen down and release it. Okay? So to get those overall large systemic changes, I have to have changes on the cellular level. So next we're going to talk about positive feedback. So positive feedback is going to actually amplify a response in a process that's happening in a biological organism. So where a negative feedback loop was turning things on and off, the positive feedback loop is going to encourage it. And it's actually going to encourage things to move even further from their set point. This would be like saying, okay, my body temperature ranges in a certain level and I'm getting too hot, and I would push it even hotter. Okay, so that's how a feedback loop is going to work. It's going to push things even further from the set point and amplify whatever response or process is happening there. So we have two examples that we're going to talk about. One we've already talked about a little bit. Okay, this fruit ripening. Okay, when we talked about endocrine communication, we talked about ethylene. Okay, ethylene again is a gas hormone. released by the plant, and it triggers the process of fruit ripening. So as ethylene is released, that encourages the fruit to continue to soften, to continue to break down its glycogen, to become more appealing to eat. Okay? But you'll notice that doesn't start and stop, right? As that ethylene is released, that encourages more and more and more. Eventually the fruit can become overripe, right? Like when you get bananas that sit out for too long and they turn all black and super mushy, because this is the positive feedback loop. I'm pushing that ripening further and further and further from the set point. So in humans, we have a couple of examples of positive feedback, both the actual onset of labor and childbirth itself, not carrying a child and not pregnancy, but the actual pushing of the baby out. So the onset of labor as well as blood clotting is also an example of a positive feedback. So with the onset of labor, with childbirth, okay, we've got a diagram happening over here on the side. So as childbirth starts, the uterus will start to contract. And as the uterus starts to contract, that will start to push the baby down. And so the baby's head will start to push onto the cervix. And so as the baby's head starts to push onto the cervix, it will start to open up that cervix. It'll start to spread the cervix apart. And as that cervix starts to spread apart, that actually sends messages up to the brain. And that tells the brain that it's time to secrete some more oxytocin. Oxytocin heads down to the uterus, and oxytocin actually tells the uterus to contract more. So the oxytocin is what triggers the uterine contractions. And the more contractions there are, the more the baby's head, the stronger and the more frequent they are, the more the baby's head pushes on the cervix, and the cervix opens up. And so as the cervix opens up, again, that's going to send signals back to the brain that says, hey, release oxytocin. So we're pushing this further and further from the set point. The set point is that the cervix is closed and that the uterus isn't contracting. And so as we push it further and further, those contractions become more forceful and they become closer together, allowing the cervix to open up completely so that the baby can fit out. Okay, so those would be two examples of positive feedback, the ripening of fruit as well as the onset of labor. So again, it's being able to apply these concepts in different places and in different scenarios, and or being able to write about a particular example of them. So we will work with the glucose insulin negative feedback loop in class. We won't spend a lot of time in class working on the positive feedback loop information, mainly because it just doesn't come up as often. And the negative feedback loop is more of a regulatory process where positive feedback is not, as a general rule. And so negative feedback is the one that we need to make sure we are 100% comfortable with.
Hi, I'm Katarina Bender, another helper of Dr. Bender. Okay, that will conclude our video for tonight. Bye!